All right, well, thanks for watching. It's pretty late here. I'll see how I do. I think I can get through this in just a few minutes of your time. So I appreciate you guys listening here. We've got the Beatles going in the background, or Beatles songs. Okay, anyway. All right, proteins. That's what this quiz is all about. So we are going to go through that. Okay, proteins. Yes, on the quiz, you're going to need to be able to list some examples and functions of proteins. That's going to be key. Yeah, so I mean, functions, you could say they function as a hormone. They transport things through our body. They are structural components. They make up our chromosomes. So yeah, examples, functions, they kind of are the same thing. There are some specific examples there listed that we've gone through: collagen, albumin, aquaporins, hemoglobin. Yes, and then there's several, all those enzymes that we've talked about are also protein. They are very large molecules. You really don't draw the entire structure of proteins, but you do need to be familiar with what an amino acid is, be able to identify the carboxyl, and be able to identify the carboxyl and be able to identify the amino. Recognize that a water is removed when two amino acids bond together in a condensation reaction, and be able to identify the peptide bond. It is between carbon and nitrogen, but you need to identify it's the carboxyl of one and the amine of the other. Yeah, same picture here. Once again, know what a condensation, I believe your book calls them dehydration reactions, and be able to look at an amino acids bonded together and find the peptide bond. The structure of a protein, it's really how do we describe its shape. The key thing here is when you denature a protein or denature an enzyme, you may change the secondary, tertiary, or quaternary structure. Really, you change those interactions between the side groups, but when we denature, we do not change the primary structure. The amino acids would still, if you change the primary structure, you've done more than denature. You've totally made a new molecule. But denaturing, it still maintains the sequence of amino acids. Hydrogen bonds hold the secondary structures together, which would be the beta pleated sheets or the alpha helices. Hydrogen bonds also hold the tertiary structure together, as well as some covalent bonds, the disulfide bridges. Quaternary structure, really it's just, once again, another way to say how are they globbing together, how are they folding into each other and stacking. It's just the overall shape of the molecule, the overall shape of the protein. Okay, same thing there. Hemoglobin, yes, how would we describe its shape? Well, it has two alpha helices, two beta pleated sheets, and they interact and fold up on top of each other. It does have quaternary structure. It's a pretty big, 64,000 atomic mass units. It has iron. Iron is a metal ion. It is a cofactor. Many enzymes have cofactors. It's really what allows them to function. Quite often cofactors or metals are where the active site is. Hemoglobin, a protein. It's really not making, well, it's a protein, and it can bind with oxygen. It can also bind with carbon dioxide. Please be able to look at this equilibrium expression and predict our shifts in equilibrium. So if acid builds up in your blood, it's going to shift the equilibrium to the right. Carbonic acid will build up, and then it will continue to shift right. And then it will basically, as long as you can breathe out CO2, this will keep shifting right. And that's what keeps you alive. Because if too much CO2 gets in your blood, it can affect the pH of your blood, because it will shift the equilibrium back the other way. And if the pH of your blood shifts, everything in your blood. If your blood becomes too acidic and that pH really changes, everything in your blood can become denatured. And if your enzymes and hormones are denatured, they won't function properly. So please, once again, if you have questions about the bicarbonate buffer system, please let me know. A great place to look is the summer packet. Go way to the beginning of the website, and there's a lot of those questions on the summer packet. I'll probably just steal from there, to be honest with you, for your own quiz. Yeah, if you add a base, if ammonia builds up in your blood, that removes hydrogen ion. And really what that does is that slows down your breathing rate. And if your breathing rate slows down, that's not good either. It's a lot of times what happens before people die is their kidneys and liver shut down. Ammonia will build up in their blood, and that will denature everything in their blood, and hormones and cell signaling molecules, really everything just shuts down. Okay, so how do we describe denaturing molecularly speaking? How or why do proteins become denatured? Well, you can denature really three ways: change pH, change temperature, or change the solvent. In my brain, it's all the same. If you change pH, so here we got a protein in maybe an acidic environment, a neutral environment, and basic environment. In an acidic environment, I think that the acid that it sits in is donating hydrogen. That hydrogen ends up sticking to the amine. The net charge on an amino acid would be a positive in an acid. Generally, we say it's neutral at the isoelectric point, which is a lot of times around neutral. And then in a basic environment, it donates, or the environment accepts hydrogen, or the molecules in there donate hydrogen, and that's coming from the carboxyl. In other words, the amino acid acts as an acid in a basic environment, and you get a net negative charge. Who really cares? Okay, so the charges change. That really is what affects those side group interactions, the coiling, the alpha helices, the beta pleated sheets. All of that is determined by these charges and those side group interactions. So if you change the charge on the side groups, you change those interactions, you change the shape, you denature the enzyme. Okay, yeah, there's another picture to see it. Okay, please be able to read these energy graphs. Just know the starting point, the stopping point. If you end with less energy in the molecules than you start with, that is a spontaneous reaction. That is a negative change in energy. That is a negative delta G. Examples of spontaneous reactions are catabolic reactions. Our peroxide, yeah, we made a mess, but you guys put a lot of water and oxygen into the room, breaking down that peroxide all day. When you added enzymes that were not denatured, it should have broken down even faster. So same thing there, just your teacher is in the picture. Okay, enzymes, yes, they really affect everything. Remember that cofactors are metals. That is really why they can act as a catalyst quite often, or oftentimes that is where an active site may be. I love to think of enzymes as they provide an environment so that reactions can happen. But if the enzyme is denatured, they don't provide the same kind of environment, and it may not function as well. Once again, keep in mind, denaturing only affects second, third, and fourth degree structure, not the amino acid sequence. How can you denature? Think of our lab. Change the pH, change the temp. We could have dumped salt in there. Dumping a bunch of different salts would have affected it as well. Yeah, and if you think of it, if you change the environment protein is floating in, you can change the shape because the hydrophilic and hydrophobic amino acids will respond differently. As we get into gene regulation, our next chapter is really all about genetics. But that's going to lead into what activates our genes, what turns our genes off. Well, that's enzymes and proteins and how they bind together. Quite often, binding can turn things on. It can also turn things off or inhibit a reaction. If it sticks right to the active site, it's competitive inhibition. If it just binds to it somewhere else on the molecule, it still could change the conformation of the active site. Just because if you stick somewhere, the whole molecule is going to change shape. The active sites may be changed as well, but it's not sticking directly to it. It's non-competitive. Yes, please be able to interpret, not do math, but just plug signs in to Gibbs, the equation for free energy, but really it's the equation for spontaneity. Will this reaction happen? Will the ice melt? Well, melting ice is endothermic. Melting ice increases the entropy, and the sign of delta H and delta S are the same. We say the reaction is temperature dependent. And quite easily, melting ice at high temperatures, at big T's, you get a negative delta G and you are able to melt ice. Burning gas, combustion reactions, those are also spontaneous. They are exothermic. You make more gas, the particles start moving around more, they become more randomized, entropy increases. That is always a negative delta G. Now what's weird is if you do this game for building proteins, for condensation reactions, a positive value minus, so condensation reactions have to put energy into them, and the molecules become less random. The amino acids become ordered. They're all a bunch of different things, and then they become one molecule. They get less random. That is a non-spontaneous event. So it's like, well, there, Mr. Gibbs, Mr. Thermodynamics. Is life possible? Well, yes, it still is. We're still here. And I thank you for still listening. You probably broke down a lot of ATP, but that provided the energy for you to build the proteins so your brain could work and you could listen. Okay, thanks.